everyone. I'm Amelia Taylor, and as she noted, I'm a data scientist at Zymergen, and I'm excited to talk with you all today about outlier detection, an inherently unsupervised problem, and how we're going to tune parameters for that. In this talk, I will give some background on Zymergen biology and robots, talk a little bit about outlier detection and tuning hyperparameters, particularly in our context, but that could work in lots of contexts, and then how we solve the problem of tuning hyperparameters in this unsupervised world. So Zymergen is a combination of zymology and genetics. Zymology is the science of fermentation to make applications or to make materials and molecules. And genetics is, of course, the study of genes and genetic variation. And at Zymergen, we combine the genetics to improve microbes to make better production. So we make new products and make the bugs faster, stronger, et cetera. And when I say faster, stronger, I always like to say that that's things like make them so that they need less food to make more product, and there are other ways you could think about that. So, of course, people have been using microbes to make things like beer and cheese for millennia. One of the first genetically engineered products was insulin, and since then we've learned how to make lots of things using microbes, everything from drugs and flavors and fragrances to coatings and plastics, and not so long ago spider silk. So there's lots of opportunity here. And programming microbes by changing their DNA to make these products is an industry worth at least $80 billion a year. So you can imagine there's a lot to do here. The market is actually served by fermentation in these really huge tanks that hold hundreds of thousands of liters of a culture. And you can imagine that it's really expensive to take a new experimental microbe and stick them in one of these tanks and see how it does, right? That's super expensive. So at Zymergen, what we do is we take, oops, sorry, that's, we do lots of small experiments in plates, gather that data, and then predict how things will do at large scale. And we can do this by massively parallelizing the process and using robots. So robotic automation drives our throughput needed to screen the large number of variants that we want to test. We see robots here moving liquid around. The middle one up there is picking out variants from a pool. They're moving plates around, and they're actually doing different types of measurements. So we do all these different parts using robots, and they move these plates through very quickly. In this context, then, our computational tasks are varied, and they include things like designing these variants, automating the data collection so that we can connect the DNA sequences to the output and the results of these tests, identify those that are statistically significant improvements, and then also predict, again, how they're going to do in these tanks. Because if you recall, I mentioned that it's really expensive to run new strains in the tanks, so we're not going to like run them through the plates and then just stick them all in tanks, right? That defeats the purpose. So we have to be able to predict what's going to happen. Quick shout out to my colleague Janet Matson, something I'm not going to talk about today, but if you want to go look, she spoke at PyCon recently about designing the large number of variants. It's a pretty great talk, and it's online. And, but our focus today is on this automated data collection side of things. So another look at our machine learning pipeline, you might think about it this way. We predict the strain designs, we build them, we stick them through this high throughput, that's the robots, they're running everything through. And then we want to be doing the rest of this very automagically. Like, so the outlier detection, then we normalize that, we can then look at that data, decide which ones were the best, use those to predict what's going to happen at large scale and to inform the next round of design. And we can do this over and over and over again at very high speeds and massively parallelized. It's pretty awesome. We have a great diverse team working on this. I primarily work in the blue boxes, so designing the plates in the factory, outlier detection, which we're talking about today, and I've also worked a lot on the question of the predictive, how to predict what's happening at scale. Here I want to make a shout out to my colleagues Flash and Nathan, who've also worked a lot with me on the work that I'm talking about today. So when I talk about outlier detection, I think we all have a general sense of what that means. But at Zymergen, we have a situation where we have domain knowledge and we know that we have contamination and process failure. And so what we want to do is we want to, in a very automated way, as I showed you, we want it in that robotic pipeline. We don't want to have a human in there looking at these and going, oh, well clearly over here on the left-hand side, those didn't grow. Right? So that's some sign of process failure or contamination. And over here on the right-hand side, we know that those values are outside of the range of how much growth we could possibly have. So those are definitely contaminated. And then you get the stuff in the middle, and you might ask, well, is that just due to the normal variation within our distribution, or are those actually outliers? So now we're bringing in some human piece of this. But most importantly, we have neither the humans nor the time to go through and look at every single data set that comes out of our factory and make this decision. It's just impossible for us. So we want to be able to have an algorithm do outlier detection, and we want it to do a good job. But you can imagine that just like any machine learning algorithm, it has hyperparameters, and tuning those hyperparameters is going to determine whether or not we even manage to pick up the stuff on the two outside edges, and how it handles these pieces in the middle. And the classic way to do hyperparameter tuning, right, is to do cross-validation. That's what everybody does. But that assumes you have a supervised data set, right? And outlier detection is inherently unsupervised. So you can't take sort of this classical approach to do it. So what do we do? Well, the approach I'm going to talk about assumes that you have some sort of outlier detection algorithm to start with. You may have your own in-house proprietary one. There are 4 great ones in scikit-learn. But you need to pick one of them. To work through the example today, I'm going to use elliptic envelope and Mahalanobis distance. I always struggle with that word. It's awesome. But the Mahalanobis, that one's one of the older ones in there. There's 3 isolation force and local outlier factor are also very nice algorithms that you might want to go check out. So what is the Mahalanobis distance? It is this lovely formula over here on the left. X is your data. Mu is the center of your data, a mean or a median. Sigma is the covariance matrix. And when you take that formula and you set it equal to a number, that defines an ellipsoid. So here in this picture, you'll see I have 2 ellipsoids. One of them is completely cut off. I'll talk about why in a minute. And it works really well in multi-dimensions. So all my pictures are going to be in 2 dimensions because we're humans and we can see those and I can graph them nicely. But there's nothing about this algorithm that restricts you to 2 dimensions. What I want to talk about a little bit here, though, is why I have one of those ellipses completely cut off. It was just too big and it didn't fit on my slide nicely. And in that case, these 2 ellipses that you're seeing have the same hyperparameter. But in one case, I'm using the maximum likelihood estimator for the mean and the covariance matrix. And in the other one, I'm using a robust estimator for both of those. And you might imagine which one is which, right? The maximum likelihood is the one that's doing a very bad job of putting ellipse around my data. And the robust one is the one that's seems like it's picking out sort of that core distribution in the middle there. Yeah?
Question, can you define for me a hyperparameter in this setting?
So in this setting, the hyperparameter is actually going to be that distance that we're putting on the Mahalanobis function there. It's this number here, and I'm going to get into this, keep going on this a little bit. That's the hyperparameter that we're interested in here. Is like, how do we choose what number we should have there to be able to pick out the right ellipse, to pick out the right chunk of distribution there. And I got like a whole bunch more slides on this, so this is awesome. So here, just to further illustrate why you really, really, really don't want to use the maximum likelihood estimator in this case. No matter what I do to that hyperparameter that she just asked about, that distance, what it does is it affects the size of the ellipse. And in this particular case, no matter what I do, you can see it hones in on that one point up there, which is definitely not my core distribution and probably an outlier. So like no matter what I do to that parameter in that context, I'm not going to do a good job, right, of picking out the right thing. But that more robust guy there is doing a pretty good job. And how do we use that? So that one's actually picked out using the minimum covariance determinant, which is also in scikit-learn, and actually the primary way in which this is implemented in there. And I put the paper reference up here, but you can also get that off of scikit-learn. It's all in there. And what this does is it takes a subset of it goes through lots of subsets of the data and picks the one with the minimum determinant. And it turns out that does a really good job of sort of picking out the core of your distribution, particularly in a case like this where you've got a real core and lots of outliers. But this still doesn't answer the question that we're interested in. We're just getting a sense of like how to draw these ellipses. We still don't know how big to make that ellipse. So the hyperparameter here is how big do we make the ellipse? Should it be the ellipse I have drawn? Or should it be any one of these other ones? And I want to pause here for a minute because some language I'm going to use a little bit is really helpful for you to think about. So when that hyperparameter is smaller, then my ellipse is going to be smaller, and I'm going to be saying lots more things are outliers. And that might be kind of way in the core of that distribution, and I'm saying lots of things that might actually be inliers or outliers. And as that hyperparameter gets bigger and bigger and bigger, I'm going to say more and more things are inliers. And we kind of want to find that magic ellipse there that is really kind of separating and picking out sort of a good separation between the outliers and the inliers. Another way to think about this, and this is kind of core to the solution, is that when you think about outliers, especially in our context where it's coming from contamination and failure and things like that, in my mind, those should really be far away, so in a sense, in space from the core distribution, right? So what we might think about for a metric is one that looks for good separation. So this ellipse here, I can see, well, it's kind of got the core distribution in there, but if you look really closely, it's maybe a little hard to see, but there are some points on the edge there. And then these next three are kind of near it. So maybe that's doing good, but maybe it's not, because it's not really quite giving me a separation between what it's going to call inliers and outliers. Whereas maybe if I blow it up a little bit, now it's got some things that's showing some variation out of that core distribution, but these points are now very well separated in space from everything else. And what we want to do is capture that in a metric, right, that we can run automatically, and then it comes out and it says, here's your best hyperparameter. Okay, so attempt number one. We used a paper by Marks, Campello, Zamek, and Sender to come up with a metric. They actually give a metric for this. And I'll go through sort of the big ideas here, and then we'll see that it was a great place to start, and some pitfalls. So I put some things on here just to sort of remind you. We're using an outlier detection algorithms out of scikit-learn. We're going to here in a minute talk about kernel logistic regression, which is also of course in there. We use lots of pandas for working with the data, and it's all wrapped up in lots of good Python love. But let's walk through sort of how this algorithm works, and how we get the metric. So the first thing you do is, like I said, you have to have an outlier detection algorithm in the start. And again, I use this one as an example so we get a sense of what the hyperparameter is in this story. But you could use any outlier detection algorithm that has hyperparameters and to do this process and to tune use this metric to tune your hyperparameters. So then once you run outlier detection, you have now labeled your data, right? it has a label. It might be a terrible label. Your parameters might be horrible, but they are labeled as inliers and outliers. And now we're going to treat that then as a supervised data set and shove it into a maximum margin classifier. You could use any maximum margin classifier, but kernel logistic regression works particularly well here because, first of all, you can think about the kernel and the shape of your data. And second of all, it gives you a set of probabilities for free. And those are actually going to form our metric. And the reason is because if you think about, again, that separation, if the parameters and my outlier detection algorithm has done a good job of labeling the data and I have good separation, then the kernel logistic regression is going to get that classification pretty spot on, and it's going to say, oh yeah, these are your outliers with really high probability, and these are your inliers, and those probabilities are going to really reflect the structure there. And if you think about it, if it's muddled, and you have all kinds of muddling and they're not well separated, then that kernel logistic regression is going to reflect that in the probabilities. It's going to say, well, I'm not really sure. This looks like an outlier, but it's not really clear. And then we can aggregate those up into a final metric. So you take them, you sort of average over the ones that were labeled outliers, you take all the probabilities and you sort of mix them all up together in that final function called the chance adjusted metric. That's what they call it, so that's what I will call it. I think it's more interesting to kind of look at what happens, but because there's a whole lot of math here. So if you go back to thinking a little bit about what I said earlier about the separation, you think, okay, so when that metric, for this particular hyperparameter, when it's small, we're kind of saying too many things are outliers, so our metric should be lower. And then as it grows, our metric should get bigger and bigger and bigger because it says that's better and better and better. And then eventually it should drop off because we're saying too many things are inliers, right? And so this seems like that's what your metric should do. And I went and I implemented their little algorithm out of their paper out of the box and started using it. And let's just say my graphs did not look anything like this. And none of that was happening. And it just was not even, it was like a big mess. And furthermore, again, we would like it to be that if I spot check it, it's intuitive. Eventually I want this fully automated with my robots, but if I spot check it, it should look right, and it didn't. So what happened, and there was a great talk this morning on keras and vocal stuff. I'm sorry for those folks, but it was good. And the part that really stuck with me was at some point they said, you should really go and look at your failures. And I thought, yes, that's exactly what happened to me too. I went and started to say, like, why is this failing? And started looking at the data in lots of different groupings. And the thing that I discovered is that we have lots of different ways of thinking about how we group our data for different kinds of distributions based on both the biology and the process. And depending on how you group them, this metric would pick out incredibly different hyperparameters. This is what we call that particular hyperparameter, but it's just that Mahalanobis distance. But it would pick out the best parameter being incredibly different depending on the grouping that you were looking at. And that's what was causing my final aggregated picture to look like a jumbled, crazy mess. So what did we do? We thought about all the different ways we could group different chunks of data based on our workflow, and this is just a subset of the possible options there. And we went back and said, okay, well, so it's a little more complicated. We need to think about all those different groupings, both for the outlier detection and the kernel logistic regression, and how it's interacting with those two things. Then iterate over those different kinds of groupings, do some normalization of those metrics, and then re-aggregate them all together. So here in green, I've highlighted sort of the original Mark's paper, and then around here sort of the general workflow that we have. But the sum up of that is think about the groupings of your data and what distributions make sense, and then be willing to kind of separate it out, iterate over those, do whatever kind of normalization is right, and then aggregate into a final chance-adjusted metric. And in doing that, then we were able to get what we expected, pick out the best possible parameters, our results are intuitive, And we're now able to do this across microbes, across processes, and we have it running in a Jupyter notebook, but we are in the process of putting it into the full pipeline. One thing I want to hit on just here at the end that a few of you might have noticed that I didn't really talk about, so how the parameters get into this process, right? If we're tuning hyperparameters, we should be feeding in lots of parameters. Depending on the size of your data, this works very well with just doing a brute force grid search. You can do that. We're working on making it work well with gradient descent, or you could do whatever of your other. What's nice about this is really about that metric, and all of these other pieces you could plug and play what makes the most sense for you. Just as a reminder, does this work for any outlier detection algorithm? Yes. There's nothing special about the fact that I was talking about Mahalanobis distance and the elliptic envelopes. And even better, you could actually use this metric to compare. You could go back to those original 4 outlier detection algorithms I was talking about, use each one of them in this process of computing the metric, see what metrics you get out, and compare them. And the biggest one should be doing the best job of picking out the separation and your outlier detection, picking out that separation in your values for outlier detection. And I'm a little bit short, but pretty good. And so I want to say thank you. A quick plug, at Zymergen we're hiring. And at Insight Data Science, which helped me transition from being an academic to working at Zymergen, their next round of applications are due August 20th. So check them out. Thank you.
Okay, we have time for questions, if anybody has one.
Your talk was about looking for an analysis of a single run, but you're doing a great many runs, and there's some similarities among them. Could you talk about how you would extract information from nearby samples?
I'm not sure I understand, but for the outlier detection, we actually tune these parameters to work across many runs in the factory. So we don't change them every time a run goes through the factory.
Got it, thank you.
Yeah, so and that's where I was talking about those groupings and the aggregating. So when we tune, we actually will take sort of at the highest, we'll take a bunch of different experiments and a bunch of when we take the data to do the tuning, we do it across all possible groupings, and then that's where that iterative aggregating process is really important, so that we get a set of parameters that are working across the whole, like across many runs and across many wells and across many plates and all of those pieces. Yeah.
I noticed that you can see.
I think you need the mic because this is all being recorded and they want people to be able to hear.
I noticed that you're using SVM to do the training. When the parameters grow, SVM sometimes will become extremely slow.
I'm actually using kernel logistic regression, which is related to SVM is another maximum margin classifier, and so you could use that also, but that's not actually what we've been using lately. Another thing to keep in mind is that I think compared to a lot of people, our data sets are pretty small, so I have not tested this on really big data sets.
I see, thanks.
But there are also, I want to just say there are lots of places in here, like my original implementation of this, there's a lot of places that it could be optimized, so yeah.
Are you concerned more with false positives or false negatives? Do they have different costs in labeling things as outliers?
In this particular context, so that becomes an even bigger issue when we think about the predicting to scale. I think about that a lot. Here, for this particular situation, there's a little bit of cost, and I would say we tend towards being okay. I'm trying to remember which one I would call positive or negative, but being okay with calling things inliers that are actually outliers. And less so the other direction. It's better for us to err towards it, calling it an inlier, because it might turn out to be the magic strain, and than the other way around. So we tend to sort of err in that direction if we're going to err, for sure. There's one here, and he's had his hand up over there for quite a while. So make sure you hit him after this guy.
Can you make your parameter selection point a little more concrete? And maybe let me frame it like this. I assume you have data that has a bunch of metadata like time of day that the sample was run, and maybe some middling things that may be relevant and may not be relevant. So how do you select the parameters you're going to include in this model from maybe a lot of metadata you have?
For this one, that's a good question. At the moment, we're trying to keep it simple and really just say, I've got a bunch of measurements, and use sort of the distribution of those measurements. We use a lot of that other metadata in other ways, and I think it would be interesting for us to consider more ways to incorporate some of that in our outlier detection. For example, like at some point, the fact that maybe we know this machine tends to fail more often, but the current algorithms that we have running, we're not incorporating that yet.
Can I make sure I understand? So you're saying that you have domain-specific knowledge of what is relevant to your problem?
What's that? Say that
You're
again?
saying you have domain-specific knowledge of what's relevant to your problem, and you're selecting based on that?
Yes. Yeah, I think that's a good way to answer that question.
So picking back on that false positive question.
Yeah.
So you're systemizing the discovery process, but a lot of scientific discoveries are serendipitous. So for example, penicillin was serendipitously discovered. So is there a chance that you could be missing like a game-changing false positive by like moving towards the systematic robotic discovery?
I think the answer to that question is probably yes. But of course, I would say our daily efforts are around making sure we don't do that. But yes, absolutely.
I know this question was asked earlier, but in your hyperparameter search, was that, what exactly was that went into the Mahalanobis distance calculation? What hyperparameter is part of that equation that was used for the initial outlier detection before you fed the results into a kernel logistic regression?
Wait. Oh, okay. So I'm not. Let me see if I'm getting your question and answering it. But so you're asking, like, in the outlier detection up here, how is that hyperparameter being used?
Yeah, from what I understood, you have at the end of the analysis a Mahalanobis distance statistic that might have been computed using some hyperparameter, and then you have a chance-adjusted metric that you compare to that.
Oh, I see. Yeah. So let me back up for a minute and go back to the simpler picture here. So I'm not actually comparing the Mahalanobis distance to the chance-adjusted metric. The chance-adjusted metric is telling me whether or not the Mahalanobis distance I picked was a good one. So I'm using the Mahalanobis distance to say, to define that ellipse, and then things in the ellipse are considered inliers, and things outside of the ellipse are considered outliers. And that's where that Mahalanobis distance is. But then what we use the chance-adjusted metric to do is that's telling me whether or not that ellipse is the right ellipse. So if my ellipse is really too small, then the metric is really small. And if my ellipse is too big, then that metric's also really small. And that sort of peak in the metric is where that distance is the right distance. Does that help?
Yeah, I'm just curious about exactly what the hyperparameter is in that situation.
Oh, okay. So in general, I had a friend the other day do a really good job of defining hyperparameters. So the way I think about it is if I go back to the Mahalanobis distance. So in parameters, so the word hyperparameter comes from the fact that to distinguish it from parameters in the model. And parameters here are things I'm going to infer from the data, like the mean and the covariance matrix. And a hyperparameter is something else that I need to keep going forward, but I'm not going to be able to necessarily, I can learn it from the data, like by doing hyperparameter tuning, but it's not something I'm inferring from the data in the same way that I would a parameter. Does that make sense? And so that's why the distance here, this number out here that's defining the ellipse, that's why that's a hyperparameter. And that's why we use that language. And we need some way to decide, like, I mean, I could, as a human, just go in and put in a that's the difference between a parameter and a hyperparameter. As a human, I can just stick that in there, whereas the parameter
Yeah.
should be something I learned from the data.
Yeah, I was just wondering if maybe it was like a shape parameter to the distribution that's being used to draw like your mean or something.
No, it's being used to draw that ellipse. It is
Okay.
specifically drawing that ellipse.
I think we have time for a couple more.
So I hope this isn't a proprietary question, but I was actually hoping you could tell us a little more about the biological side of this.
Maybe.
Yeah. What, or if you can, like, what were you mutating the genome itself and just seeing which one would help hurt the bacteria grow? What kind of system was it, and what were you trying to produce? And just anything.
I can't say anything about what we're producing or what bugs we're in, but yes, that's what we do. We change the DNA. We make DNA edits and see what happens. Yep.
Are you mostly editing the genome itself, or are you editing the plasmid, or is that? Yeah.
Matt,
sorry. Okay. Sorry.
Matt, what are we doing? No. Genome itself. Genome itself. Thank you.
Maybe one more?
Call on the bioinformatics people in the room to help out.
Hopefully the easiest question you've been asked so far. I was curious about the dimensionality of the hyper ellipsoid that you were working with. So how many dimensions were you solving this problem over?
I don't know that I want to answer that question.
Is it
It
tens,
varies.
hundreds?
Oh, no, it's smaller than that, more like tens and smaller. But it varies depending on the particular data set that we're looking at too.
Right, I was curious about the
Yeah,
scalability.
yeah, it's pretty, it's not
into very large
hundreds.
dimensions.
Yeah.
Okay, thank you.
Okay, maybe one more?
Hi, I was wondering if you could just describe a little bit more that iterative process. So, like, how do you do those groupings? Yeah, I just wondering if you could just elaborate a little bit more about.
I would say the best answer is we're using a lot of domain knowledge and sort of really engaging with our biologists and thinking about, like, when we think about how the bugs behave depending on the particular measurement that we're worried about, what levels would we expect those distributions to be sort of a single distribution versus maybe they're all behaving with different distributions. And so we use that sort of internal in-house proprietary, or just like domain knowledge to think about that.
So is that part still automated, or?
Making the decision about how to group them is not automated. But then once we decide how to group them, the rest of it, then this is all fully automated. Yeah.
Okay. That's all the time for questions. So thank you, Amelia.